Hello and welcome here in Constance or wherever you are. I'm going to tell you about the ASPA and I'm illustrating how it works based on a case study. So I hope this is okay, that this will be on long distance, but of course I invite you, if there are questions, discussion points or others, to send me an email or to address some of my colleagues. So I'm sharing the screen with you and you get a little bit of an overview how it looks like here, but we will dive now into the ASPA and I will show you how it can be applied with a case study asking whether there's a DART or DNT liability for picoxystrobin. So what's the ASPA? It's something you know very well, but also something that has innovative elements. So we all know that for next generation risk assessment, we need building blocks like a problem formulation, toxicokinetics, and then the integration risk assessment. But these are very coarse building blocks. So this is a more fine-grained, more organized structure. And this is the ASPA, and we will come to learn about this more. It's also implemented in a software which is already done, available to some, being beta tested, and which will be released next year. So in very short words and as a simplified structure, the ASPA is the alternative safety profiling algorithm, an algorithm because we follow a defined structure. It starts with the problem formulation. The problem formulation can be adapted, and that means decision points will depend on the problem formulation. And then there are main blocks like hazard, ADME, and exposure. They have some connections. For instance, within the ASPA, the hazard module, there will be screens and hit follow-ups. There will be in silico technologies, identifications of mode of action, definition of a point of departure, and then this will interact again by the use of an in vitro to in vivo extrapolation to give input to the data integration. And this is, of course, an essential part of the risk assessment. So these are the course blocks, and it's a procedure. All those that have thought more about next generation risk assessment will be well aware of. But let's go more into some details. So IATA is something you know, but it also takes the concept to a new level. So what we need here is, of course, transparent input requirements. They need definition, reliable selection of tools. This is where it starts getting a bit more difficult. A defined data generation within these tools, and then solid decision rules, when to go on, when to go into more depth, when to exit, when to enter other modules. And then, of course, reliable data integration procedures, not just throwing all data on the table and seeing what one could do with it. So that means we want to have all this to get confidence into the predictivity, the reproducibility, and the relevance, and especially reproducibility of the outcomes at the given input. And what is not assured with the present available technology with the rough blocks is that different groups of assessors would come to the same conclusion, that the same type of data would give the same outcome. And this is the innovation in ASPA. We want to make sure and more that this gives the same outcome, that we have a quantification and also quantification of remaining uncertainties. So as an evolutionary process, we could imagine a conventional IATA, so with the modules of information, they would be kind of converted with time into an algorithmic IATA with defined processes within and between the modules. And then in the end, there would be a really formalized tracking and documentation of all these steps. And this is what the software is doing that I presented in the beginning. So just to give you some idea and an overview, these would be some of the large modules, and each of these modules would have sub-modules. Just to give you an idea, the hazard identification could look at targets, it could look at genotoxicity, but then there could be whole batteries for certain purposes. There could be questions, is there any metabolite toxicity? Is active transport related to the toxicity? Of course, there could be a read-across module, and linked to this, there could be in-depth follow-ups, so AOP confirmation, secondary assays, consideration of time aspects. And of course, there could be very dedicated batteries designed for specific data gaps, like the developmental neurotoxicity in vitro battery on top, or extended hazard testing to increase specificity, to get more information on potency and so on. So the overall structure would be that there would be certain modules with tests or integrated test strategies, and then sometimes there would be a decision point, and depending on how the decision is taken, yes or no, there would be kind of a reiteration or an exit of the ASPA. So just to briefly exemplify how such a decision point would look like, we can pick again hazard identification. So one question could be, is the collected information sufficient? Then if this is not, then it would bounce back and more assays, more data would be required. And this would need to be parameterized. So is the collected information sufficient is not a clear question. So we need increasing levels of guidance on the criteria to be considered for this, on the data to be used, on the threshold of effect, on the threshold of certainty we want to have, on consistency checks with previous decisions, and so on. And this would all be documented within such an ASPA process. And an alternative decision could, of course, be there is enough information, and then one could exit the module towards another module or to the risk assessment. Good, so let's move on from there with a case study with a focus on DART and developmental neurotoxicity. So the goal is to test how well the ASPA elements are suited for hazard characterization of developmental neurotoxicity. And the second goal is to define gaps and develop improvements of the ASPA. So this is why we want to run a number of different case studies. So this one starts off with picoxystrobin, which is shown here. And there is kind of the active structure, that's the strobin structure. So picoxystrobin belongs to the strobilurins, which are used as fungicides. And the compounds and their metabolites may remain in the environment and are taken up mainly through the oral route. So this is important to know as a background for the problem formulation, which is the exposure of the fetus in the relevant range of an adverse outcome in humans? Or as a second question, does the contaminant represent a risk for the European consumer regarding DNT, developmental neurotoxicity? Or can the workflow be extended to other strobilurins? And that's basically the outlook and the future of this case study. So picoxystrobin is one of many, many strobilurins, and therefore we will look into this one compound, and at the end we will ask the question, could we continue and use what we learned here for the other strobilurins? So we would start, as always, with the problem formulation. This is the standard IATA approach. And we have seen what the problem formulation is. And then the next step, the next module would be collect existing data. And then there would be a decision point. Is the information already sufficient at this point? If it is sufficient, if there's loads of information around, one could define points of departure, one could do risk assessment. If the information is not sufficient, then one would move on to the next step. And the next question would be, is read across applicable? And there is a read across module in the ASPA. So if read across is applicable, one could exit with yes and enter this module. If read across is not applicable, which is the case here, we would move on. And moving on means entering the big modules of exposure, of ADME, and of hazard. And I'm going to show you how this could look like. So first we start with the different ASPA tiers, and the big modules like the exposure module would ask for an estimate of the external exposure. And of course, one needs to know the use scenario for this. So the use scenario is intake as a food contaminant. Then there would be a calculation of the external exposure. And here's an exemplification using, for example, the highest level would be the acceptable daily intake. There could be also studies on food consumption and others. So we used here as the maximum level of exposure a value of 0.09 milligram per kilogram. And that can be used to calculate internal concentrations. Of course, that's not a trivial procedure. So we have defined the exposure. Then we would come to the internal exposure by using in silico and in vitro barrier models. That would give input to a PBK model, and the output would be expected concentrations in different tissues and the plasma. And in this special case, because DNT and DART are at question, there would be not just the maternal tissues, but also fetal plasma and brain information. And that would also be derived from special ADME modules. So all this would be an algorithmic procedure with different decision points, step by step, and with clear definitions how the different steps are being done. Good, just to come back, these are the big modules to get everybody on one plane again. And of course, the modules have connections. So the connection between ADME and exposure is the internal exposure. One of the connections between hazard and ADME is knowledge on whether there are perhaps hazardous metabolites or special transporter dependence. This was just a reminder because now we enter the ADME module, so one would have an estimate of the external exposure. And then the question would be, are the ADME properties of the compound known? If they are not known, then one would need to generate basic ADME data. So that would be absorption information, information on barrier transition, information on clearance, and that information could be used to run a PBK model. And this is the output for picoxystrobin, for instance, the maternal plasma concentration at the highest possible intake for population distribution, or the fetal brain concentration. Of course, many of these data can be generated in the PBK models. So in silico PBK modeling to derive maternal and fetal plasma and brain concentration has been done and shows what this big module can do. Now we move on to the ASPA modules. And first we would go for general hazard alerts. So there would be an estimation of hazard using first in silico tier. And for this, QSAR can be considered. We have a pharmacophore. This could be input in different models. And actually it was done here. So we did in silico docking to different targets. And the compound docks very well to complex 3 of the mitochondrial respiratory chain. So this gives a potential target which can be followed up. So there's an alert to also enter a specialized in vitro battery for developmental neurotoxicity, as we know that a number of mitochondrial toxicants are also developmental neurotoxicants. Next level would be a NAM screening. There's a whole screening battery available to get broad information on what kind of biological activity the compound has or could have. And finally, these data would need to be integrated. So this would give alerts to enter specialized in vitro batteries, for instance, for DNT, if there's an alert for this, or to go on more with the hazard characterization. So the next tier could be, for instance, after the data integration and there's sufficient alert, to ask, is there a specific AOP triggered? The answer could be no. Then some broader screening is required. The answer could also be yes. Then the question is the information enough? So is the AOP detailed enough? Is there sufficient information on the triggering of MIE and key events? If the answer is no, the outcome is the same as if there's no AOP, because then more specific testing is required. And this could be done in the developmental neurotoxicity test battery, which has been established here, and it consists of a large number of assays that all screen for key neurodevelopmental processes. And the outcome of this data of this battery would be used for data integration, and then further decisions can be taken. And I will lead you a little bit more into this. So for instance, the picoxystrobin testing was done in different assays. And the difference is, in most assays, it was not a specific hit, it was cytotoxic. But in the UKN2 assay, which tests for neural crest cell migration, there was a big offset between inhibition of migration and viability. So there was a hit here, and the hit was followed up then. So then the question is, are the concentrations from this assay relevant for the hazard characterization? Can we determine a point of departure? And sometimes this requires a number of follow-ups. So different follow-ups could be secondary assays, looking at migration in other ways, so that gave a hit confirmation, or mechanistic assays. For instance, if something is a mitochondrial inhibitor, it should block the oxygen consumption. It also does this in 2 different assay setups. And of course, the assays can be varied to kind of approach better the physiological situation. Here we have a longer term exposure in a different medium. And the big difference is we get to the 100 nanomolar level of potency. So this is important for defining the right point of departure. And now, taking this information, one can ask, is the nominal concentration the relevant one to work with? And one can do measurements in cells, or one can do in silico modeling. So we use the VIVD model of Sotara, and this is the outcome. So if 1 micromolar picoxystrobin would be added to a culture dish, then we would expect something like 84 micromolar within the cells or the mitochondria. We would expect something less in the media. So we see about 100-fold accumulation of the compound in cells. So the nominal concentration would need to be corrected to get to the right tissue concentration. So this was then also considered, and I think with this I have exemplified all the different considerations within the ASPA. So the final point is, can we compare the points of this departure to the exposure, and can we get to good displays that make this data transparent? And here the display shows the different exposure levels, for instance, modeled for the fetal brain or modeled for maternal plasma. So we get to different concentration ranges. And this would be synoptically compared to the different adversity levels. So we have seen under different assay conditions, we get to different adversity levels, and the most sensitive assay was in the 100 nanomolar range. And now we can do ratios between exposure and adversity, and we can look for a margin of exposure. If we then also take into account that the nominal concentrations are not the same as the tissue or cell concentrations, the final outcome is the margin of exposure, or the most sensitive assay, and the highest exposure level is at least 80-fold, and that can be the basis of the risk assessment. So I hope this gave an overview how the ASPA works. So in the end, one would exit the ASPA with a documentation of the risk level and the different uncertainties with a as transparent as possible display. And in the end, we get to some learnings and an outlook. So we feel the ASPA may be used for D&T hazard assessment. There are two different entry points. So either the specific problem formulation, as in our case, was for D&T, or the pre-screening would give alerts to enter such a module. And one could also work towards quantitative AOP to put this on a more solid basis. So such an AOP could look like this. So it takes also into consideration the time of exposure, the accumulation in cells. And of course, this could be probed with other strobilurins. So with this, I'm ending. A lot of people contributed to this. So it was a case study from Risk Hunter. It was discussed at the D&T meeting. Of course, a lot of people in the group in Constance provided data for this. And I hope I could give you an overview, and I invite you again, if there are questions or remarks, just to contact me or one of my colleagues. So I wish you a nice day or a nice evening whenever you're looking.